the kind introduction, and thanks to the organizer for the invitation. And it's a great pleasure for me to share with you about some of our recent work. So I'll start with this perhaps counterintuitive statement that the genomic sequence might be the most underexploited resource in genomics. So what I mean here, right? So of course, genomic sequence is widely used, an important tool in all of the genomics research. Right? First of all the times when we are using the genomic sequence, it doesn't really involve understanding the information that is encoded in the sequence, right? Or when, for example, when we are aligning a read to the genome, or when we use it to detect mutations, right? We're using genomic sequence more like a map rather than reading it like a book. And secondly, considering how much information is encoded in the genome, or how much biology is already encoded in the genome. So the amount of information that we can extract is still a tiny, tiny fraction of that. Right? So considering how most of the biological discoveries were made, most of them were made without the need to understand how the information is encoded in the genome. Right? So there's a huge gap here. It is both a great challenge and a great opportunity for us to be able to better understand our genome and able to extract that information for discovering new biology. So understanding the genomic sequence, I think, is a almost perfect setup problem for machine learning and AI. Right? So of course, this is because we have lots of genomic data sets that are already available to us. So we have those genome-wide measurements for many, many different activities about genomic sequence, from transient vector binding to 3D genome organization to transcription, and across many, many cell types. And we have new assays emerging very quickly. Right? And for those data sets, they are a very valuable resource for understanding the genomic sequence, but they don't directly tell us. They don't directly tell us how are those activities encoded in the sequence. Right? Or in other words, how does sequence determines those activities? And the sequence determinants of its activities are something that we can capture through computational modeling approach, like building machine learning, deep learning models. Right? So I think there is a huge space of questions that we can ask by developing computational approach to capture and exploit the dependencies between sequence and its activities. Right? And in my lab, we typically think of those directions as three major parts. So I call it prediction, understanding, and design. So prediction is the most well-developed direction, and understanding and design are more newer and nascent questions. So for prediction, we ask a question of, you are given a genomic sequence, can we predict its activity from just the sequence? Right? Or if there is any mutation to the sequence, can we predict the impact of that mutation on its activities? So I've been working on this direction for a bit more than 10 years for now, and sorry for the format problem. And we've developed a bunch of computational methods for predicting from the sequence to different kind of activities that measures about the chromatin and 3D genome and transcription. Right? Of course, many others have also contributed to these directions. And a lot of times that we can make models that are making pretty good predictions. Right? And it is beyond our current knowledge of biology can explain. So in those cases, we think that those models has also captured some hidden biology that we have not yet understood. Right? So by studying how those models work, I think it's a great new approach that can help us understand what is the underlying mechanism of sequence-based regulation. So that is going to be the focus of the talk today. And finally, the third direction on design is a flip side of prediction. So instead of asking the question of, given the sequence, can we predict its activities, we can also ask, if you want a certain type of activity, how do we design the sequence that can give us that activity, right? So that wouldn't be the focus today, but actually I'll tell you the story that touch upon all three of these directions. So it'll give you a taste about what I mean for all three of these three questions. So let's start with a story. So it begins with us asking a very basic question, which is, what is a promoter? Or what should a promoter sequence look like in the human genome? So we all know that the promoter is one of the most basic elements in the genome, right? So for a gene, you have to have a promoter, right? You can have a gene that doesn't have an intron or doesn't encode a protein, but without a promoter, it won't be transcribed. So all genes have to have a promoter, right? So it's arguably one of the most fundamental elements in the genome. However, you are asking the question of what should the promoter sequence look like? What should it look like in the human genome? We actually didn't have a very satisfying answer to that. So over the past few decades, people have identified a bunch of called promoter motifs across different organisms. And those motifs are only explaining a small fraction of human genome promoters. So if you look at an average promoter, so even if it is a highly expressed promoter, a lot of times it has none of those motifs. For example, the famous TATA TATAA motif only appear in no more than 10% of human genome promoters. So for vast majority of them, if you are looking at the sequence and we ask the question, why is it a promoter and why transcription start at the position that it starts at? We actually didn't have a very satisfying answer to that. So that is a question that we want to address. And to do it, we start off by first casting it as a machine learning question. Right? So we want to ask, so given the genomic sequence, can we predict this base pair level transcription initiation profile? So what is shown at the bottom is for a single transcript start site, if we plot exactly where transcription start from and count how many times it starts at each location, we get a transcription initiation profile like this. Right? So for most genes, actually for all genes, transcription doesn't just start at one base location. So you start within a region about 50 to 100 base pair. Right? And if you plot how frequently it starts at each location, you get some profile like this. So the wiggling profile that you see here, the wiggling is not noise. So it's actually highly reproducible if you do the experiment again. And it's even reproducible across different cell types and tissues. So as long as the gene is expressed, you have a very similar transcription initiation profile at the base resolution. So we think this profile is actually reflecting the underlying mechanism of transcription initiation and of how the transcription initiation machinery was assembled at this promoter. Right? So if we can explain how does this transcription initiation profile shape depend upon the genomic sequence, then we might have captured the key element of how sequence organize transcription initiation. So with this idea, we first built a deep learning model that can make accurate predictions of this transcription initiation profile from sequence, taking about 100 kilobase pair sequence as input. And actually, we're not the only one who had this idea. And also, Anshu Kandaji's lab from Stanford and Charles Dunkel's lab at Cornell also built similar models at around the same time. So what's unique about our approach is that we're able to analyze this deep learning model and use what we learned to build a very simple model that only have a small number of parameters. Actually, it's so simple that we can interpret every one of its parameters, and only it also take about 600 base pair of sequence as input, but still achieve almost the same performance in being able to predict the transcription initiation profile. So that will be the focus of the talk today, and just first show you these the performance of the model. And these are some example predictions. of this of the simple model called puffing and the deep model puffing D compared with the experimental measurement. Right? So if we quantify this performance by correlation, and we can see that so for both the simple model and deep model, you can achieve very high correlation, especially for genes that we can well measure, which have high coverage. And both of them are getting even higher correlation compared to the correlation between two most similar experimental measurements between phantom cage data and encode cage data. So this doesn't mean that the model is perfect, but it certainly shows that the model has captured something important about how the sequence determines transcription initiation. Right? So how does the model do that? It only involves quite simple computation. I would say only 2 critical steps of computation to predict that. And the first step is that we look at the genomic sequence and we scan for motifs. And in this case, the motifs are learned just by training through machine learning approach. We didn't give it motif to start with. And after the training, the model can detect the locations of motif in this sequence, and different motifs are colored differently here. And the second step of computation is that we just need to convert each of these motifs into its effect. So each of this motif activation curve is now converted to a motif effect curve, right? And that is almost all of what we need to do. And we just need to sum up the motif effect at every location. And that is almost already our final prediction. So we sum up the motif effects as well as the effects of 2 other simpler types of sequence patterns. And then we just need to rescale it. And that already makes very good prediction for the transcription initiation profile for most human genome promoters. And to show you for more than one gene, here is a movie where we captured the prediction for many human genome promoters. And as you can see that the location and the types of the motifs are very different across different promoters, right? But with this simple rule, you can predict most of its transcription initiation profile very well just through the motif combination of motif activation and motif effects. So what are those motifs? And so this slide is probably the most important slide of all, which captures all the motifs and its effect on transcription initiation. So all of those motifs were learned just through training with the sequence and the transcription initiation data. Right? So, but after we have learned the motif, we realize that most of them are actually no motifs. Right? So, but except for Tata, perhaps, most of those we actually didn't know whether those motifs would directly contribute to transcription initiation or how would it contribute to transcription initiation. And those effects were plotted on the left. So how to read this plot is that if we position the motif at the center, so for example, if we look at I don't know whether I can show the pointer. Yeah, we look at the Tata effect. We place the motif at the center. We can see that the Tata will activate transcription about 30 base pair downstream of that motif. So instead, for YY1 motif, you will activate transcription immediately upstream of the YY motif. And also it has a second weaker effect about 200 base pair upstream of the YY motif. And we can capture the effect of a motif on both the positive strand and the reverse complement strand. And the reverse complement strand effect was plotted in this dashed curve. So we can see that the Tata and YY1 are both very strand-specific motifs, but you can also see a bunch of motifs that have almost symmetric bidirectional effect, which means that it activates transcription in both the forward and reverse complement direction in almost the same, with almost the same effect. Right? So, but still, every one of these motifs has a distinct transcription initiation effect curve if you look closely. So for example, you look at Fy, it shows this very strong 10.5 base pair periodicity. So 10.5 base pair is also the helical periodicity of DNA, which means that it rotates back to its original phase every 10.5 base pair. Right? And we think that this indicates that the Fy might have a more rigid interaction with the POL2 pre-initiation complex, which make it showing this stronger periodicity, and because it has to stay at a certain surface relative to POL2 pre-initiation complex. And this also shows a little bit for the other motifs like SP, but not as strong. So we think each of these transcription initiation effect curves are reflecting the underlying molecular mechanism of how does this motif activate transcription. And I hope that we might learn more about it once this structural basis of how this motif activates transcription is understood. And lastly, there are two special motifs here. So U1 SNRNP motif is also the 5' splice site motif. So we see it here, or we know that it's actually having an effect only after transcription initiation. So this is because that we can see this effect if we model the transcription start site of total RNA transcript, like using the cage data. But we don't see it if we model the transcription start site of the nascent transcript data, like ProCap. So which means that this 5' splice site motif is enhancing the amount of transcript after transcription initiation happens. So that's also consistent with recent data that shows that the 5' splice site U1 SNRNP motif is promoting the processive transcription elongation. So instead of having an effect on transcription initiation. And finally, the long initiator motif was the only motif that we didn't recognize at the beginning. But if we look at it closely, we realize that actually it is a motif, it's an elongated version that combines multiple previously known co-promoter motifs. So all of those motifs were thought to reflect the affinity of TF2D, a part of the pre-initiation complex. We also think that the long initiator reflects the preference of TF2D on transcription initiation. So it fine-tunes exactly which base the transcription will start from. So now, other than that, we can show that we can actually accurately predict transcription from these effects. Can we also directly just validate that those effect curves that we observed are correct, right? Actually, can we show some experimental evidence that these are reflecting the true effects of these motifs? So because this models is fully explainable, so we can do something like this. So we can go into the model, and then we ask the question of, what if we remove one transient factor from the system? Right? Can we turn off one transient factor and see what's going to be its effect on transcription initiation? Right? So for example, we can ask, if we remove an Fy, what's going to happen to transcription initiation? So we can just set the motif activations to 0 and set the motif effect on an Fy to 0 and predict the change of transcription initiation after we do that. So because most of these motifs activate downstream and repress upstream, so if we remove it, it shifts the transcription initiation site toward upstream. So if we plot its effect genome-wide, we see that for promoters, we have strong NFY contribution. We see the transcription start site shift toward the upstream. Right? So this is a prediction. And fortunately, this experiment has already been done by a different group. And we look at their data, we see that the experimentally observed effect of NFY knockdown is almost exactly consistent with the computational prediction from the model. And we can also do that for another transient factor, YY1. So if we do the in silico knockout of Y1, this is the predictive effect of Y1 in silico knockout. And we can compare with experimental data. It also shows a very similar effect compared to our prediction, right? And also even shows a little bit of the weaker effect of Y1, that's 200 base pair upstream. And beyond this, we can also test the model on the effect on deleting and inserting motif, right? And on this example, if we delete or insert some certain motifs, we see that the model predictions is highly consistent with the experimental measurement as well. And we can see this effect actually even without any experimental data. So if we look at the evolutionary conservation of those motifs, we actually can already see hint of how this effect contributes to transcription initiation. So here, in this plot, we center at the transcription start site, right? And we look at the conservation of different motifs depending on its location relative to the transcription start site. And we can see that the Tata motif is most conserved if it is 30 base pair upstream of transcription start site, so where it has the strongest effect on transcription. And we look at the YLM motif, it's having the strongest effect just immediately downstream of the transcription start site, which is also where it has the strongest effect on transcription initiation. And we can even see this periodicity of NFY by just looking at this where it has the strongest conservation relative to the transcription start site. Right? So the evolutionary conservation information is already informing us about this motif's position-specific effect. And so this has given us a lot of confidence about the effect curves that we have captured are real. So now we release the preprint, and after that, actually we have also seen even more experimental evidence supporting what the model has captured. So for example, one of the motifs that we were surprised to see and actually wasn't known to contribute to transcription is ZNF143. So ZNF143 was actually thought to be a looping factor, right? So, and this paper from Anders Hansen's lab studied ZNF143 as a looping factor, but they found that actually has no looping effect. And instead, it has effect on transcription, right? Just exactly as our model has predicted. And this other work has also verified that for those motifs that we predicted to have an activating effect for downstream and repressive effect upstream, they actually indeed have that effect, have that repressive effect towards the upstream, which wasn't previously known. So all of those are very consistent with what the model has captured. So, and the final test that I'm going to show, which wasn't published yet, is that if we really believe in the model as capturing the rules of the transcription initiation, then we should be able to create new promoters by following the rules that are given by the model, right? So we should be able to randomly combine those different motifs that should contribute to transcription initiation and use them to create new promoters. And that's exactly what we did. And indeed, we can just flexibly combine any of those 10 motifs to create new promoters. And the created promoters transcription initiation profile is also exactly consistent with what the model has predicted. And we can also predict the activity level of this of the promoters that we designed. So these are these rules are not only capable of explaining the human genome promoters, we can also use it to design new promoters with the activities that we want. So now I think we are quite confident about the rules that we have captured. At least it explains a big fraction of how sequence determine transcription initiation, right? So understanding this, how does it affect our understanding about the gene regulation, right? So we can now analyze for every human transcription start site what are the motifs that contribute to this transcription start site, right? So here we choose that the motif contribution for the top 100 transcription start sites in the human genome. So every column here is a different promoter. And among those top 100 promoters, Tata has a disproportionately large contribution, but we still see contribution from every single one of those motifs, right? So they contribute to some of these promoters. And we look across the genome about almost all of the human transcription start sites. We see some of the motifs contribute to a different subset of the human genome promoters. So all of them are responsible for some of the promoters' activity. And SP is the most frequent, most common motif, but all of them have contribution to a subset of promoters. So what are the differences can we observe from the promoters with different motif contribution, right? So we actually see a pretty striking effect when we are looking at these promoters expression pattern across tissues and cell types. So in this plot, what we plot is the expression level and tissue specificity of every transcript. So each dot is a transcript. The x-axis shows the expression level of that transcript, and y-axis shows the expression dispersion of the cell type specificity of that transcript. So every plot shows the same dots, but they are colored differently based on different motif contribution. So for example, here we can see that, so for the transcript of transcription start site, I have a strong Tata contribution. It is prone to have more tissue-specific expression pattern, which is also previously known. But we also see a different preference for tissue specificity versus ubiquitousness for every single motif, especially for Y1, it strongly prefers a ubiquitous expression pattern. And all the other motifs have some degree of preference to ubiquitous versus more tissue-specific expression pattern, whereas the U1 SNRMP, also the 5' splice site, is neutral to the transcription, which is expected as it has no effect on transcription initiation. So why is that, right? So none of those motifs are highly tissue-specific. None of them bind to highly tissue-specific transcription factors. Almost all of them bind to some transcription factors that are ubiquitously expressed. So it's not because they prefer expression in certain tissue. And we think that this is reflecting that these different promoters has a different dynamic range. So the Tata promoters could have a higher dynamic range and it's more sensitive to transcription activation signals. So there's a lower baseline and can be activated higher when it is activated by, for example, an enhancer. Whereas the Y1 promoters have a lower dynamic range. So it has a higher baseline, but it's less affected by transcription activation signal. And so this is also consistent with our computational experiments where we use a deep learning model to test what are the expression level if we insert each promoter in different locations of the genome. So we see that the Tata promoters are more selective at which locations it is inserted into. So it's expressed in fewer locations, whereas the Y1 promoters are less selective. It will be expressed in more promoters, so in more locations that we insert it into, right? So this is our hypothesis. That's why these promoters are reflecting the tissue specificity of the genes, and I hope that we will see more evidence toward or against it in the future through the experiments. And another aspect that we can explain with the model is why the transcription usually happens at both directions in human genome. So if you look at the transcription initiation activity for most human promoters, actually we see both activities in the forward direction and the reverse complement direction, right? So in the past, there has actually some debate about whether this means that the human genome promoters are inherently bidirectional or they are inherently directional. Right? So one of the questions that this debate hinges on in the end is that whether these two directions of transcription actually depend upon the same sequence, right? Whether the same sequence is leading to transcription on both directions. And we think that we can answer this question now. Just qualitatively, I think you have already gotten a hint that it is because that we have those bidirectional motifs that's activating transcription at both the forward direction and the reverse complement direction. Right? So bidirectional motifs appears at almost every human promoters. So it is leading to transcription at both directions at the transcription initiation level. And those directional motifs, and especially UN, SN, RMP, will enhance transcription, the mature transcript at one direction, which leads to more mature transcript on one direction of the transcription. If you measure at the mature transcript level. Right? And we can also quantify this effect quantitatively. And for this promoter, we can quantify at the base resolution how much each base contributes to transcription in the forward strand, and how much each base contributes to transcription in the reverse complement strand. And the contribution is also colored by the motif type it is mediated through. And we can see that these contributions are mostly shared. And actually, this is true for most of the human promoters, which have which activate transcription at both forward and reverse complement direction. And if those two transcription start sites are within about 300 base pair distance. So for most of those cases, we can now say that the sequence that are contributing to both directions are mostly shared. So finally, we can look at this, how conserved the rules that we have captured are across different species, right? Because we also have very good measurement for mouse, so we can train the model on the mouse data and use it to predict on the human genome, or we can train the model on the human data and make predictions on the mouse genome, right? So it turns out that the predictions are almost identical. And in fact, if we use the model that is trained on the human genome and make prediction on the mouse genome, it's basically the same performance as if we train the model on the mouse data and predict on the mouse genome. So we can say that there is not any distinguishable difference that we can see between the transcription initiation rule between human and mouse. And what about beyond human and mouse? So that question is much harder because we don't have the transcription initiation data for most of the species, but we try our best to look into these sequence conservations. So previously I've shown you that if we look at the sequence conservation of those motifs, it shows stronger effect at the positions where it has a stronger effect on the transcription. It's more conserved when the motif at the position that has a stronger effect on transcription initiation, right? So that is from the human genome's point of view. If we repeat this analysis from the point of view of every one of these mammalian genome, and we also see very similar conservation curves, which indicate that they likely also have a similar transcription initiation effect for those motifs. Right? So we think that it's likely also conserved across all the mammalian species. So with this simple model, by being able to build a very simple and explainable model, so we are able to actually learn a lot more than what we could by just building a deep learning model. We can use it to dissect how each motif exactly contribute to different promoter activities. We can understand the effect curves of each individual motifs, and we can stratify the promoters based on the contribution of different motifs and look at their differences in expression pattern. We can explain the directionality of these promoters and look at the conservation across species. Right? So I think promoter is still one of the most basic and perhaps the most simple regulatory element in the genome. Right? And there are still a lot more of genome activities that we couldn't explain easily from the sequence level. And I think this approach is also totally generalizable for us to understand all the different kind of activities in the human genome. And finally, to look into the future, I think I want to just revisit this graph that I showed at the beginning. I think I'm very optimistic about what we can do with sequence modeling, because there is still a lot of synergy that we haven't exploited among those different directions. So, for example, I've shown you that with a good predictive model, we can analyze the predictive models and try to use it as a tool to understand what are the sequence-based mechanisms of this transcription of the underlying regulation, right? And if we have a better understanding, we should be able to use that to design better predictive models with better performance and generalizability, right? And understanding will also allow us to do better design, like what we showed that we have an explainable model for the promoter. We can use those rules to design new sequences. And validating these designs allow us to verify our understanding, right? So to make sure that our understanding is correct, or prove that it is incorrect or insufficient. Right? And the predictive models can be turned into design models. So we have a predictive model. Now we're developing methods that can allow us to make designs for any activities that we have a predictive model for. And also developing design algorithms can also help us doing prediction, because we can actually unify both prediction and design through a generative modeling framework. So for all of those directions, we improve on one direction. I think it simultaneously helps us do better on the two other directions. So I'm hopeful that in the future, by advancing all three directions, we'll substantially improve our ability to decode and understand the genomic sequence. And finally, hopefully, we can come back to the initial question that I posed, that we can make decoding the genome a major driver for biological discovery. So finally, I would like to thank all the people who have contributed to this work. So Sinia is the first author of the puffing paper, and Donghong contributed to the experiment that validates the model, and Chen Lai developed the website. And this is in collaboration with Jian Zhou lab at St. Jude Children's Hospital. So thank you for listening. I'm happy to take any question.
So we have time for some questions. Thank you.
Thank you. That was really beautiful. So I'm interested in bacteria, which I think are, humans are kind of dull. But no, so there are many groups of bacteria about which we know very little experimentally, and we're sort of warped because there's this view we intuitively end up thinking that all bacteria behave like E. coli, which isn't true. Do you see any directions that your technique could be pushed in areas where there isn't experimental data or isn't much experimental data to do the training?
So that's a very interesting question. Actually, I'm also very curious about what are the rules of how the sequence determines transcription in bacteria. So actually, so in a human genome, there is a slight advantage because we have bigger genome. We have more training data to train the model. But I think because we can now do it with a simple model, it might not actually require a lot of training data. So I think with the relatively small amount of data, it's still possible to be able to generate those models. So I don't know before we try, but I think that's something that I'm definitely interested in. And also for bacteria, I'm also curious about how different these the sequence rules for different bacteria are, right? So if they are more or less conserved across similar species, then we can also pull the data from related species to build a model. But it will be also very interesting to look at the differences in different species. So, yeah.
Yeah, and to some extent, it's not the same proteins. It's not just not the same motifs, but the proteins that compact the genome, for example, are different outside of proteobacteria. So it'll keep you busy for a while.
Yes.
So our lab's been looking at, we've been observing some patterns of nucleosome positioning sequences with some dinucleotide every 10 base pair like sort of patterns. And I was wondering, outside of like transcription factor motifs, have you guys been able to see like more degenerate sequence patterns that maybe like traditional logo finding type approaches weren't able to, identify?
Right. So, so there are some other, I mentioned there are some other simpler sequence patterns in the model beyond those motifs. So one of them is what we call trinucleotides. So there are some mysterious things in the model that we still don't fully understand. So one of them is that these motifs are not sufficient. So we also, in this case, we also have to include the effect of trinucleotide or dinucleotide. Trinucleotide is slightly better than dinucleotide, but it doesn't necessarily mean that biologically it is trinucleotide. It's just a small simple sequence pattern we can use to capture the residual sequence dependencies that are not captured by motifs. So could it reflect some of the nucleosome positioning preference? I think it's possible. Another hypothesis is that it could be related to this DNA methylation. That's also another direction that we're exploring. So the trinucleotide has the highest effect on transcription initiation are the CG-containing trinucleotides. And also we have initiator motifs, which will contribute mostly not to the activity level of promoter, but exactly which base pair that the transcription start from. So I think that's less likely to be directly related to nucleosome positioning, but possibly some of the effects are related to the, it could be reflected in trinucleotide. Also, each of these transcription factors, they may have its individual effect on positioning the nucleosome, and those effects might be having, so those effect curves, right? So some of it might be related to how does the transcription factor position the nucleosome, which in turn affect transcription.
Would it be worth like trying to expand to like 10 base pairs, or is there already like connectivity built into your model between the trinucleotides? Like I'm surprised that di to tri improves that. We can talk more about it.
Yeah. So for nucleosome positioning, it is a much longer. We think of it as a motif. It's a much longer motif.
When
right?
10 base pair exposure of the minor groove around the nucleosome, I wonder if 10 might be the window where you.
Yeah, it will be interesting to explore that.
But we can talk more about it.
Yeah. Thank you.
Hey, yeah, this was super fantastic. Yeah, really cool to see what you've been working on. One thing that just occurred to me from this last question is, what about like DNA shape? Like, could the di and trinucleotides be giving you information about like shape motifs that are maybe not captured in like, or that is like tougher to capture in the motif like letters themselves?
Yeah, we haven't tried to incorporate motif parameter, but that would be very interesting to test. I think some of it is partially captured by sequence, but some of it which is not captured by sequence, I think we have to test whether including the motif or the shape scores would help in improving the prediction.
Right. Cool. Yeah, I think Lydia Fredolino has some nice and also Hassan, forget his last name, but he's also been working on they found that shape motifs do actually give you a little bit extra that the sequence doesn't capture. The other thing that I was thinking is that like the motifs that you've, because we're talking about promoter architecture, I could imagine that might be fairly conserved. And so I was wondering if this allows you to maybe say something about promoter evolution across like maybe primate history. If you could like look at other, kind of like closely related to human species, do you see these same motifs, and can you like see like gain and loss maybe over time? Like could be informative about like why you see like particular like motifs, preferentially end up in certain types of genes as well.
Right. So we think that the sequence rules are conserved across all mammals, and I think among primates, it's likely highly conserved. So if we look at the sequence level, so it depends on what TSS we are looking at. Actually, I haven't looked lot on primates, but between human and mouse, there are many promoters that actually sequence has already been very different. So many of them are nearly identical, but others are very different. So we can certainly see the gain and loss of motifs across species.
Yeah, that's really cool. I'm thinking about those like, human accelerated, like kind of regions, if any of those are in promoters, and if you could explain them using that type of architecture, for example.
Yeah, it will be very interesting to see the evolution of promoters through this model's perspective. Yeah.
Cool. Thank you.
Thanks.
Hi, Dr. Zhou. Thanks for sharing this impressive work. So I have a question about the Yuan's nerve. So you included 10 or 10-ish transcript factors, but this one does not look like a typical transcript factor. So what's your consideration to include this one in this study? And another question is more specific. Looks like the effect of the Yuan's nerve is more in the upstream of TSS. You mentioned that it's like post-transcription effect to enhance the TSS downstream transcription. So it sounds like kind of controversial. So do you have any explanation about that?
Sorry, I didn't hear your first question clearly. Sorry, can you repeat that again?
Yeah. So why include Yuan's nerve? Because it sounds like why include U1 as an RP?
Okay.
Yeah, it sounds a lot like a typical transcript factor. Yeah.
Right. So UNSRP doesn't affect transcription. So actually, we see that if you only train on the nascent transcript data, you don't see UNSRP. So it only show up when we train using mature transcript data. So which means that it has effect on transcription level, on the transcript level, only after transcription initiation. So that's consistent with what people think it does. It's enhancing the processive transcription elongation. So it makes us produce more transcript that doesn't affect transcription initiation.
Okay.
And for the effect that activate downstream and repress upstream, I think it could be just reflecting that if you have this motif here, it tends to position the PO2 toward this position relative to the other than the upstream position, right? So if you have PO2, it will be shifted toward downstream. So it's probably more of a shifting effect other than this real repressive effect. But indeed, this effect can be experimentally observed. So if you have you insert a SP motif to the downstream of the TSS, it actually indeed repress transcription.
Okay. Thank you.
Yeah, thanks.
If I can ask a question here, very nice talk, John. It's the mouse conservation or the mouse prediction test. If you condition that on the degree of conservation, do you see different performance levels, like a highly conserved regulatory region versus one that's not conserved at all? Are you able to predict those equally well? Because if so, that suggests you really understand you really have the fundamental rule. Or can you comment on that?
Yeah, that's an interesting question. I haven't really stratified the promoter by its conservation level. We sort of just concluded that this we can apply human model to mouse and obtain identical performance. Actually, we can obtain higher performance if we use a human model rather than the mouse model on the mouse genome. But we probably have a higher resolution of looking at this if we focus on the sequence that have diverged. And yeah, since if the sequence is identical, it's expected that it have similar prediction. So,
But if you can predict those ones that are not at all the same, equally well, that really confirms you understand the rules, right?
Yeah, I still believe that it is we'll still see the same observation, but that will be something that would be good to look at.
Maybe I'll just add one more question since we have a couple of minutes. One thing that I think is really exciting about your work is how much you learned about mechanism after you built a model that was working. I think a lot of us are applying machine learning to different problems. And I think that's a really productive process you went through. So I'm wondering if you could can generalize that. How do you approach a problem like that? Once you get a model working, how do you unpack that to get back to mechanism and learn something? Because for many of us, yeah, it's great we can predict things, but ultimately we want to unpack it and learn things about biology. So, which I think you do really well. So can you generalize that and talk about how you approach that for us?
Right. So I think there are a couple of things I think are important for being able to use a model to probe more about the mechanism. I think one of it is that the model should have a causal knowledge about what is happening underlying the prediction. So in this aspect, having sequence-based model have some advantage because sequence is more likely to be the cause rather than the consequence of these downstream regulatory activities. Right? So most of the time, the model's predictions are causal, which means that we can actually see this effect if we actually mutate the sequence and experimentally see the effect to be consistent with the prediction. So I think capturing the causality is one of the key to begin with. And then I think a lot of times that we just build those deep learning models or other black box machine learning models, right? So I think because underlying biology is often very simple, we can try to understand this machine learning model and try to figure out what to use to make this prediction and use that to build simple models. So these are, for us, that process typically start with just doing what we call like a virtual genetic screen, which means that we introduce perturbations to the input and see how it affects the output. So it is kind of like how geneticists study the genome, right? So we just introduce mutations or design screens to test a certain hypothesis. We kind of do that in the same way. We can do unbiased screens by just mutating every single position of the input sequence, or we can have some designs that focus on specific, for example, combination of different motifs and to test whether their relative positioning have an effect. So by designing these virtual genetic screens, we can use it to probe what the model has learned. And once we think we have a good understanding about what the model has learned, we can build that in to design those simple models. Right? And designing simple model is still a trial and error process. It's not necessarily what you think at the beginning is best reflecting the data. So you still need to see how well that simple model can fit the data and also just try different designs and to see which one ultimately is both simple and has the good, can faithfully capture the data. And I think by combining these two approaches, we can build more, ultimately have simple and explainable models for almost everything that we can predict through deep learning models right now.
Maybe one last question.
It's short. I just want to make sure I understand this correctly. You're using conservation only as a way of validating. You're not using it as an input at all.
Right.
Is that correct?
Right.
Yeah. So that can give you another potentially explanatory and also, that could be an input angle.
Right. So I think if we use evolutionary conservation, we can just highlight the bases that are more likely to have an impact. Right. So especially in the case where the data is limited, I think that will be helpful for the model to learn meaningful motifs. Yeah.
All right. Thank you, John. And we have